This is Jose Merino, Editor-in-Chief of the Neurology family of journals. The Neurology podcast provides practical information to neurologists and other clinicians to help them provide better care for their patients. Thanks for listening and have a great week.
Hello, my name is Gordon Smith. I'm a neuromuscular neurologist and chair of the Department of Neurology at Virginia at Commonwealth University in Richmond, Virginia. I'm excited today to have the opportunity to talk to Jordi van Unnik, who is a PhD student at the University Medical Center in Utrecht, and Ruben P.A, who's an assistant professor and medical statistician at UMC Utrecht in Utrecht, Netherlands. Doctors Van Unnik and Van Eijk are going to talk with us today on the Neurology podcast about their article, Development and Evaluation of a Simulation-Based Algorithm to Optimize the Planning of Interim Analyses for Clinical Trials in ALS. It's a long title, but a really exciting article. Jordi and Ruben, thanks so much for joining me on the podcast today.
Thank you very much for the introduction.
This is going to be a really fun discussion, and it's at a really, I think, pivotal time in ALS. Obviously, we've had a couple of new drug approvals, most recently tofersen, which is exciting. In fact, we did a podcast recently with Dr. Suma Babu from Mass General about this approval, so our listeners might want to check that out after listening to this. Despite this excitement, of course, most clinical trials in ALS and, for that matter, other degenerative disorders have been negative over the years. I wonder if we can begin by telling our listeners what problem you've tried to solve in this study. What were the goals? What were you trying to achieve?
As you highlight already, so the main aim of the study was to think about ways of how can we identify studies that are not going to show any results, so futile clinical trials. How can we identify those upfront or sooner so that you can stop such a study and then hopefully invest the remaining resources that you have and the remaining time that you have into another drug development line. So that's the main aim of the study. And related to that then is what would be the ideal timing. So if you're doing a study, so what would be then the ideal timing to look at whether the drug has shown the intended effect or whether it's already time to conclude the futility.
So, Ruben, there must be a number of benefits to ending a study earlier when it's appropriate to do so because of futility, beyond just saving money and budget. I mean, can you talk a little bit about the other reasons this is so important and objective?
Of course, there's like the time and the money component, but especially for ALS, it's for patients maybe the most important reason. So they want to try out treatment. So if we know already that at one point the treatment is not going to be effective, then the patient has much better chances to switch therapies and try something else out. Another reason is that some drugs, unfortunately, had some safety issues. So what we show in our studies that we actually applied it to 2 trials with severe safety concerns. So there was evidence that there might be a beneficial effect, but in the end, during the trial, it turned out that there was actually a harmful effect of treatment. And I think that that's very important to identify those kind of drug effects very early on and make sure that we have enough evidence and make the right decision to stop such a study. And in that way, prevent doing harm to our patients.
Yeah, Ruben, I really appreciate that answer. I'm just thinking about the context here in the United States. Of course, we have the Healy platform trials going on, which are really exciting to patients because they have potential access to multiple therapeutics. And I don't know, as an administrator, I often tell people, let's fail early and fail often. If you're going to fail, get it out of the way and move on. And I think this is a really exciting approach. So, Ruben, thanks for that great introductory answer. And I wonder, Jordi, if we might just begin our conversation, and we're going to get into more detail momentarily, with what the answer was. What did you find? How did this simulation-based algorithm work? Can you provide a high-level summary for our listeners? And, maybe talk a little bit about your thoughts about how this is going to inform clinical trial design. And I think this really lays the foundation for the rest of our discussion.
What is currently done is that the statistical analysis in most clinical trials for ALS are performed after patients have completed a fixed follow-up. And some clinical trials include interim analysis that are performed after a pre-specified number of patients have completed a follow-up. And in these instances, pre-specified timings together with their decision rules are established to determine when a trial may be stopped early, so either for efficacy or for futility. And these criteria in current trials are often defined arbitrarily and frequently ignore information that is available from earlier trials. So as a result, the trial design may be suboptimal. So what the algorithm does is that it tries to optimize the planning of interim analysis for clinical trials by using re-information from earlier clinical trials. And so by applying this algorithm, We show in our paper that we could reduce the trial duration and the number of patients randomized of all 3 trials that were evaluated by the algorithm. And we believe that the utilization of this algorithm in the design phase of a clinical trial can lead to a better use of time and resources available, but also minimize the exposure of patients to ineffective or harmful drugs.
That's a great explanation. And obviously, it's a complicated approach from a modeling and statistical perspective. I think you really provided some clarity for our listeners who want to learn more. And I hope that's basically all of you. I'd refer you to figure 1 in the paper, which provides a really nice schematic of the simulation-based algorithm that Jordi just walked through and gives you a sense of the impact of modeling the uncertainty of a particular agent. And what I'd like to do is maybe talk about a specific example in a moment. But I wonder if we could step back a little bit and talk more about how interim analyses are currently used in ALS clinical trials. For instance, do all ALS clinical trials currently use an interim analysis or not? And when they are performed, what's the general approach now? What's our current baseline?
So the methodology behind interim analysis that has already been invented, I think in the 70s or 60s even. So it has been around for very long, and there are many statistical papers that show doing an interim analysis is very effective. So you can shorten your trial duration and save patients being randomized to inferior treatment arms. Despite that this knowledge is well known, only about maybe 5 to 10% of the ALS trials is actually using interim analysis. For me, that's very surprising because the probability that the trial on ALS, or maybe we can generalize it to any neurodegenerative disease, is going to be successful is very low. So yeah, we did once a systematic review, and there we found this low number of trials that are implementing interim analysis. Well, we could have saved a lot of resources, I think, if every trial would implement them. And probably the reason why investigators are a little bit hesitant of using interim analysis is related to their kind of scary statistics. So they require multiple testing, and you need to adjust for that. And they often require specialized software. So there's a little bit of unfamiliarity among clinicians with them. So they're often in the lead of defining the protocol for clinical trials, and what they don't know, they often don't propose, or what they're not familiar with, they don't want to implement for some reason. And I think you often see this especially in the pharmaceutical industry. So the academic trials might be a little bit more liberal. But the other issue is that conducting an interim analysis brings some operational challenges, especially for the statistician, but also for the whole research team, because you need to close a database and you need to make sure that all the data has been input into the database. So you need to take all the charge from the patients, and then research nurse, whoever is inputting the data, needs to complete that into the data set, and then the statistician needs to analyze it. So it's quite some work of conducting an interim analysis. So I think that's one of the reasons for it, especially now with electronic healthcare records, and almost every software package now can generate a interim analysis design for you. So I don't think those borders should limit now trials of using it more, because in the end, using an interim analysis can really improve patient outcomes and also improve the use of resources.
I'm just reflecting on this a bit, Ruben. There's a human element here too, I think, that you talk a little bit in your paper in terms of the design interim analysis, or really design of clinical trials. And I think clinical trialists are eternal optimists. We never want to give up on things. And so I've sat on my fair number of data safety monitoring boards and data monitoring boards, and no one likes stopping a trial. And I think you brought a really good reason why we should be more comfortable stopping trials. I think what would be really helpful for our listeners is to talk a little bit about one of the examples. And what I really liked about your article is you developed this methodology, and then you kind of validated it by looking at 3 prior ALS clinical trials, so a couple of diaphragmatic pacing trials and ceftriaxone. And I wonder if we could talk about the ceftriaxone trial and just about how you applied the model. And so if I understand correctly, the way you did this is you looked at ceftriaxone as an agent and thought, okay, this is unlikely to be a home run. And so you inflated uncertainty around the estimates of outcome with the treatment. And then you projected a survival impact, which I was kind of curious about, because you projected would seem to me a pretty big survival impact. So 28.9 months in the placebo versus 43.4 in the ceftriaxone treatment group. And I wonder if you could just talk a little bit about, at a high level, this example, how you made these sorts of decisions and kind of what the outcome was for this particular trial and modeling your approach.
So when we design a trial, then usually we have already some kind of preliminary evidence. So often it comes from like a smaller phase 2. And this is the case what also was in the ceftriaxone trial. So that there was a seamless design, but we pretended that it had like different stages of the development. So what they first did is they conducted a smaller phase 2 trial, I believe in 30 or 40 patients. So they randomized them to placebo and then to the active drug, to ceftriaxone in this case. And then they observe a treatment effect. And then based on the treatment effect, they're going to start to design their phase 3 pivotal study. And what we often do is that we just select the treatment effect that we observe in our phase 2, and then we just copy paste that to phase 3, and we assume the treatment effect to be effect. So if we observe there like difference in the survival time, as you indicated, then we just copy that treatment effect onto the phase 3, and then we do the sample size calculations and the duration of the study. But the treatment effect is not given. There is actually a lot of uncertainty around that treatment effect. Else we would not do a phase 3 trial anymore, because else we would already be sure that the treatment would be working. And that's exactly what we did in our study here. Is we tried to see what kind of evidence did the phase 2 trial actually provide. So indeed, it did show a beneficial treatment effect, but how much uncertainty in terms of, for example, a 95% confidence interval was around that observed treatment effect. And then based on that uncertainty, we can then see what is then the probability that that treatment would show an effect in phase 3. And if it's going to show an effect, when would that effect be then? And this is what we try to approach with the simulations. And then on those simulations, then we evaluated, so if this is our uncertainty in the treatment effect, when would the best timing be to conduct an interim analysis? And if you simulate it many times, you can, in a sense, get like the best design or the best planning of your interim analysis to stop the trial as most efficiently as possible. I will give Jordi's time to reflect on that as well.
Yeah, thank you. So just to come back to that uncertainty a bit. So what we did is, rather than using a fixed estimate, we defined an uncertainty distribution, like Ruben said, did not make the treatment effect a fixed estimate, but rather follows some sort of distribution based on the evidence that was shown in a phase 2 trial, for example. But we then took it a step further, because we saw that even when the evidence in a phase 2 trial, for example, is really strong, we still see that a lot of trials fail in their phase 3 subsequent trial, so to say. So because of this, we just figured that ALS is a really difficult disease to study, and that it has a really high probability of failing a clinical trial, independent of the evidence that was shown at the phase 2 trial.
Yes, I think that's interesting that you mentioned that indeed, because that's something that happens independent of the ceftriaxone trial. So you have two sources of evidence. So you have the phase 2 that can give you some information about like how certain are you about the treatment effect. But we also know that doing any trial in ALS, there's like a poor probability of success. And that you can just estimate by seeing so how many trials were conducted in ALS in the past and how many of those were successful. So based on that, you can also have some kind of probability of success. So what, like Jordi said, is that we combine those two estimates into like one uncertainty distribution about the treatment effect.
That's a great explanation. I think it really brings into focus what you were trying to achieve and how you went about it. I wonder if we could talk a little bit about timing of clinical effect. I'm hot off the tofersen podcast, which was very interesting and a very exciting therapy. And one of the things I found interesting about tofersen is that during the clinical trial, there was a strong biomarker signal, but the clinical signal didn't become apparent until the open label extension, and those data are available on the FDA website now. How do you integrate thoughts and uncertainty about timing of clinical effect? And it'd be interesting to see how your model would have performed looking at that clinical trial, right? Because there, it may have just been a matter of timing. That's an issue. Is that one of the variables? What are your thoughts about that?
That's a very good example. So what you see in the tofersen is indeed like what we call like a delayed response. So the separation between the two treatment arms happens after 6 months, that it really becomes apparent. So if you would conduct like a interim analysis after, let's say, 3 months, then indeed it would look like the trial is futile, and then you may falsely stop. So that is indeed the risk of doing interim analysis. But yeah, that risk is in principle not different than from a design where you don't do any interim analysis. So that risk is balanced there. But I think that is really a realistic scenario also for ALS, because usually it takes time, and we can see it in tofersen, but we can also see it in other drugs for ALS, or Riluzole, for example. It just takes some time for a biological response to translate into a clinical response. It's unlikely that if we give a treatment today in an ALS patient, that tomorrow we can already show like a slowing of the disease. Probably it's going to take, well, indeed, like 3 or 4 or 5 months. So that's something we didn't really incorporate into our design, but it can be quite easily extended just by saying, well, we define like a minimum time duration that we want to see follow-up of the patient before we're going to start doing interim analysis. So I think that's the easiest way to implement it. But also on the technical side, I think that we can modify the simulation models while saying, well, we know like the uncertainty around the treatment effect. We know the probability of success in ALS trials, but we also know from few trials how the treatment response would appear if there is a treatment response. So that third level of uncertainty into the simulations might also be another strategy.
It seems to me, Ruben, that you use not only estimates of certainty or probability of a treatment effect, but data from preclinical and early clinical development, knowledge about the pharmacology and the target of the drug. And I think this is going to evolve over time, isn't it? The more we learn from hopefully more successful trials, and you'll be better able to design the right algorithm.
Exactly. So now we picked only one outcome. In our study, it was that we looked only at survival. But yeah, if at one point, so in case of tofersen, there was like a very strong biomarker response. So if the biomarker becomes a very important outcome, that maybe we can also do like early interim analysis based on the biomarker. And if we don't have enough evidence for futility or efficacy at that time point, then we switch to the clinical outcome, that you make it even more adaptive, the strategy.
You brought up the term adaptive, and one of the things I found really interesting in the discussion on your paper is how this approach might be used for other purposes. So you talked about, for instance, modification of eligibility criteria. And ALS is a highly heterogeneous disease, as implied in our earlier conversations and as our listeners know. And I wonder if you could talk about other ways this type of approach to interim analyses might be useful. And it seems to me, this seems tailor-made to integrate with adaptive trial design or Bayesian trial design. I wonder if that's the case, and maybe you can blue sky how clinical trialists can use this approach in innovative ways.
Yeah, so we agree that this is only the beginning. During our simulations, we use the interim analysis only to see if the trial could be stopped or not. But like you said, we can use the results of interim analysis to do many things. For example, to enrich the trial by changing or specify, at least more restrictive with the inclusion criteria, to only include the patients that we see an effect on.
So I think that especially for ALS, that latter part is very important. So we see that probably genetic subgroups are responding better to certain therapies. So we see now the evidence of tofersen, but there's also other trials in ALS that might show a different response among certain gene carriers of ALS genes. So another adaptation during an interim analysis could be to just decide, okay, I only continue with, let's say, the C9orf carriers in ALS, and then I drop all the other patients. So that I think that kind of adaptations is what you meant as well, right?
Yeah, I did.
Well, guys, thank you so much. And I just want to encourage our listeners to check out this article. It's a really interesting read. And I think Jordi and Ruben, you've done a great job of not only describing the approach in a way that's understandable, but the implications of this. And as I've pondered it, I think there are really great implications, large implications, not only for ALS clinical trials, but how we can apply this across therapeutic development. And I think what's really exciting about it is not just stopping for futility, but in a world where we have an explosion in therapeutic development, there's a real premium on deciding when you're going to stop a trial, because there are lots of options for our patients who really want to be part of discovery and have access to novel therapeutics. So congratulations. truly appreciate you taking the time to talk and make this understandable. And I'll encourage our listeners to check out the article. It was just published in the journal Neurology. So Jordi and Ruben, thank you so much.
Thank you for having us.
Thank you very much for the invitation.
This is Stacey Clardy, your podcast editor. Thank you for listening and for letting us join you on your commute, while you're exercising, or even while you're brushing your teeth. This has been another episode of the Neurology Podcast, your best source of practical, relevant, and timely information for neurologists, clinicians, and patients.